Hello, good morning everybody. I'm really honoured and delighted to be here. It's a fantastic opportunity to speak to new generations of psychiatrists and to tell them about the work that we're doing, thinking about how we can actually use the genetic information that is coming on board at a rapid pace in my field at the moment. I'm a psychologist, and the type of treatment that I've worked with particularly is psychological treatment. A lot of the principles I'm discussing apply equally to pharmacological treatment, which I will also mention from time to time. But one of the facts about psychological treatment for anxiety and depression that really interests me is that whilst they are effective in treating these disorders, and around 50% of people recover from their anxiety or depression following treatment with a psychological treatment, another 50% do not. So that's really quite a high level of individuals who've gone through the experience and time and commitment and expense of a treatment only for it not to work and for them to remain where they were at the beginning. And so a major part of what my research group are doing is trying to understand who responds to psychological treatment and who does not, so that we can improve the chances of helping people get into the right treatment for them from the outset, and helping clinical researchers understand what they need to know about what's preventing people from recovery, so that they can hopefully improve and develop new treatments. And so the approach I'm taking to this we've called Therapygenetics, and we literally mean using genetics to understand the outcomes following a psychological treatment. So can we use genetic information to predict variance in how individuals respond to the experience of a psychological treatment? So why am I looking at this with regard to anxiety and depression? Well, I've worked with these disorders for about 25 years, and I'm really delighted to see that they are finally getting the attention that they need from funders, from the public, from the media. And the reason they're so important is because they're so prevalent. They affect around a quarter to a third of individuals at some point in their lifespan. So that's a huge proportion of the population. And they start very young. Anxiety disorders, half of them start before the age of 11. So that's before our children have finished primary school. Half of individuals who are going to experience an anxiety disorder during their life will already have experienced it at that stage. That's very young indeed. They show huge chronicity, these disorders. They often recur time after time. They go on into adult life, where they have major impact on people's capacity to maintain normal working elements of their working lives. So they're associated with being out of work, and they're associated with all sorts of other social difficulties. And this is a projection of costs for 4 major psychiatric disorders by the year 2027. And the dark blue part of each bar refers to direct costs, so treatment costs primarily. And the paler part of each bar refers to indirect costs, so costs due to, for example, loss of earnings or taking up benefits of one kind or another. And you can see that the cost of anxiety and depression is huge. These are billions per year. That's billions with a B on the beginning. So that's 26 billion per year by 2027 will be the cost of anxiety and depressive disorders in this country alone. So it's absolutely massive. Of course, schizophrenia and bipolar are also extremely severe and important disorders, and they attract an enormous amount of research and research funding. And they also cost a lot in terms of direct costs, but their indirect costs are far lower because they are so much less prevalent. So it's incredibly important that we better understand how to treat individuals with anxiety and depression, and hopefully in time we better understand also how to prevent these disorders. Now, as you may well know, there's been a massive change in access to psychological treatment through the NHS in this country in the past 10 years through an initiative called IAPT, Improving Access to Psychological Therapies. And this works by providing treatment in a variety of ways, all of which have to be evidence-based. And they're broadly speaking grouped into 2 categories. Low-intensity treatments, which could mean an online treatment, it could mean a group, it could mean being sent some work packages to work through at home, or even doing some behavioural activation, which can be quite effective for mild depression. And then there's the standard classic high-intensity treatment, so one-to-one with a therapist. Obviously, there's a major cost differential between these 2 routes, but we don't know very much about who needs which. So this is a very important question. Who needs the high-intensity treatment, which is so much more expensive and often more time-consuming as well? So many people refer to the severity of the initial presentation as being important, which it is for psychological treatment response. The more severe your presentation, the less likely you are to benefit from a psychological treatment. And so for the more severe patients that come into IAPT, if possible, they are allocated straight to the high intensity. I say if possible because a lot of these decisions are also pragmatic. But that's really the major variable that's being used. Not a lot else is entering into the equation right now. And we don't know a huge amount about how to decide who out of a group of people is the one that really needs that high-intensity treatment. We do know a bit about predictors of poor psychological treatment overall. So as I've already mentioned, more severe presentations are less likely to have a good outcome. Those with a more chronic form of the disorder are also more likely to have a poor outcome. Those with comorbid disorders, so for example anxiety also presenting with depression, are more likely to have a poor outcome, and those living in severe hardship are also more likely to have a poor outcome. In terms of predictors of antidepressant response, interestingly, severity of symptoms is not relevant there, and you're equally likely to respond well to antidepressants regardless of the severity of the presentation. But it can make a difference if you've tried medications before. That can be useful in predicting how you'll respond to other medications. And then different types of symptom profiles appear to be associated with differential response to different antidepressants. So, for example, difficulties in decision-making is associated with better response to some antidepressants than others, similarly BMI. And there are a lot of clinical variables that appear to be associated with response to one type of antidepressant and not others. But again, the picture is not sufficiently clear to be useful, especially in clinical practice. This is data from Rudolf Uher's group, where he demonstrated a couple of years ago that by combining information about a range of different factors, you can start predicting enough variance to say something useful about treatment response. So this is an R-squared from a model, and you can see that the total R-squared for this model is around 10%. And this is for prediction of escitalopram, this is for prediction of nortriptyline. And this is combining, these are the highest loading effect, variables and their effect sizes. And you can see that by combining these variables together, you're improving the capacity to predict outcomes. And this is a figure from the same paper, and this red line here indicates an effect size that is of clinical utility. So these are various different models combining different types of variables together and indicating that by doing this, you're starting to get towards something that is of clinical meaning. But in terms of thinking about who should have psychological treatment versus pharmacological treatment, we still don't know a great deal. We know that for those who are married, psychological treatment tends to be a better option. For those who've had severe life events recently, CBT or psychological treatment can be a better option. Those who have failed to respond to an antidepressant often then do better with psychological treatment. And interestingly, those who are out of work do better with a psychological treatment. In terms of those who do better with antidepressant response, the main predictor seems to be personality disorders. So what I want to add to this picture, and this growing picture, it is growing all the time, this is a very, very active research area, and what I want to add to this is genetic influences. My background is in using genetics to understand the development of symptoms over time and how genes interplay with the environment in the development and change of symptoms. And I think of psychological treatment as one type of environment. So here I've summarised some data on anxiety, depression, psychological treatment response and antidepressant response. And we've got data from twin studies in the pale grey bars. So you can, and these are the proportion of variance in the population due to genetic influences, so otherwise known as heritability. And the twin heritability for anxiety and depression, these are just rough, is around 30 to 40%. And there's decades of research supporting that, and I'm happy to answer questions on it. I'm not going to spend long on that. These are data from two of the most recent papers on anxiety and depression taking a genomic approach, and I'll tell you a little more about this in a moment because it's from my group. So this is with 32,000 cases, 82,000 controls. We estimated heritability of anxiety from individual genetic variants, all added up together to be around 22% of the variants. The most recent Psychiatric Genomics Consortium paper from the depression group estimated heritability at around 10%. There's one paper that estimates overall heritability of antidepressant response, and that comes in at about 40%. So it's likely that the heritability of a psychological treatment response will be in this same sort of range. We call these heritability estimates moderate, so in the 30 to 40% range. So this is the data I mentioned briefly just now. It primarily comes from UK Biobank, which is a fantastic resource for people interested not just in genomics, in all sorts of things to do with medical research, because it's such a huge sample. So this is our genome-wide analysis of the anxiety data from UK Biobank. And for those of you not familiar with reading these charts, these dots are individual genetic variants, and there are over 500,000 of those on here, so it's a huge experiment. And they're arranged by chromosome from 1 all the way up to 23, and the X chromosome at the end here. And these two lines refer to genome-wide significance and trend level. Now, because you do so many tests in a genome-wide study, you have to do this incredibly massive Bonferroni correction of correcting for 1 million tests effectively. So the genome-wide significant p-value is 5 times 10 to the minus 8. And so the reason it's taken us quite a while, it's now around 15, 18 years since we've had the potential to do these genome-wide studies, the reason it's taken us a while to gather up a sufficient head of steam to get useful data for disorders like anxiety and depression is because they are affected by a huge number of genes, each of extremely small effect size. So you need these very, very large sample sizes. And as you can see down here, this is a very, very large sample. It's 32,000 cases and 82,000 controls. As I say, primarily from Biobank, then also a subset from RCPsych, the Danish study, and a subset from Angst, which is an anxiety consortium. And this is the first piece of data that we've worked with in the new Psychiatric Genomics Consortium Anxiety Group, of which I'm one of the chairs, and hopefully there will be many more papers like this to come. This is a gene-wide analysis. So the previous slide showed you what happened when you look at each individual marker one at a time, but you can also do the analysis to look at genes one at a time. And then the significance level is a little bit different. It's a little bit less stringent because there's a smaller number of tests that you're doing, and you can see that a number of different genes, and several of these are already of interest from the literature, come up above the genome-wide significant level. So this is very exciting work in the anxiety field, and this is the first time we've really had access to this type of data. In terms of thinking about how to use genetics in predicting outcomes, I thought I'd show you this new paper, again from Rudolf Uher's group, which has just been published in Scientific Reports. It's a predictive model for response to antidepressants, and it's escitalopram and nortriptyline. And this is data from GenDep and a number of other pharmacogenetic studies that have all been combined together to try and give a sufficient sample size. And what he's done here is build a model including a range of variables selected from this list here. So you can see there are some demographic variables, there are some clinical descriptors, particularly the symptom profile is broken down in a lot of detail for this analysis, as is previous medication, and then over 500,000 genetic variants from a genome-wide analysis were also included. And what you can see here is that he's built a model derived from escitalopram response, and he's built another model derived from nortriptyline. And these were done from a sample where people were randomly allocated to one or the other treatment. And the first model was built to understand good response to escitalopram, and the second to nortriptyline. And then when he tests them, the escitalopram model is a far better predictor of escitalopram response, and the nortriptyline model is a far better predictor of nortriptyline response. So this gives a first indication of where this field might be starting to go, which I think is really exciting, and it literally just came out in the last week or two. So if you're interested in this area, this is probably the most relevant paper to have a quick look at. In terms of the work I described that I've been doing about psychological treatment response, This is work that took several years to undertake, and it included recruiting children with anxiety disorders from basically all the leading clinical academic groups doing CBT for child anxiety across the world. There were 12 different groups, 20 different sets of patients from different treatment trials or from treatment as usual, and we had 3 different treatment groups in there. We had a guided self-help group, which, because they were children, was primarily led by the mother guiding the child. There was group treatment and also individual treatment. Our genome-wide analysis of that data set wasn't nearly as exciting as the one I showed you earlier because the sample size in the end was only 1,000. We had 2,000 DNA samples in the lab, but we needed this longitudinal data on all of them. We needed data before treatment, after treatment, and at follow-up. And this is one of the things that makes it harder doing genetic studies of treatment response, because you also need this longitudinal data. But it's also one of the fantastic things about doing this type of work, because you are then prospectively collecting symptoms and looking at the extent to which genes are important in understanding change in symptoms prospectively over time during the course of a known, predicted, planned environmental influence. So it's also a way of looking at the idea of gene-environment interaction. By the time we reduced our sample to those who had sufficiently good quality genome-wide data and had longitudinal data, it was around 1,000, and you can see we have nothing at genome-wide significance, a few at the trend level. But we did another analysis with this data, which allowed us, and I should have said this controls for all the things you might expect to be relevant, so site, gender, the primary severity of primary disorder severity, treatment modality, and what the primary disorder was. But what you can also do with this data is create polygenic scores, and effectively what you do is do a huge regression analysis and then identify which are the genetic variants that are contributing significantly to your prediction, and then you make a score out of them weighted by their beta weight. And so, for example, it might be this portion of the distribution, and then that gives you a single score that tells you about genetic influence on the trait that you're interested in. So one of the traits that we were interested in exploring with respect to psychological treatment response was something called differential susceptibility. And this was an idea that had come out of the candidate gene literature, which I'm not particularly telling you about today, but happy to be asked about it later. The idea behind differential susceptibility is that rather than thinking of genetic vulnerability as purely vulnerability to a negative response, genetic vulnerability or genetic risk might instead be acting as a way of making individuals more or less susceptible to the environment. So some individuals may be very susceptible to the environment, and then they'll do extremely well in good environments. So like this orchid, they'll bloom if they're looked after very well. But they're quite difficult to look after. They require a higher level of care than others, and if they don't receive that, then they do particularly badly. So these are just highly sensitive individuals who can be contrasted against what we think of as dandelions in the analogy, who will thrive anywhere and for whom the environmental influences have less of an effect. So in another study that we had been doing, this is with my colleague Rob Kears, we had created a polygenic score that captured this idea of differential susceptibility, and we decided to have a look at that in our treatment response data set. And I'll just walk you through this figure. So these are individuals in our data set who had a very low score for genetic sensitivity to the environment. These are individuals who we would expect to be less responsive to the environment, both positive and negative. And at this end of the distribution, we have individuals who are highly responsive to the environment. So these are people that we think will do very well in good environments, but will also struggle a lot in poor environments. And then I mentioned previously that we had 3 levels or 3 modes of delivery for the psychological treatment: the high-intensity individual CBT, group, and then parent-led CBT with a workbook. And what you can see is that for the individuals who had the very low polygenic score for response to the environment, it really didn't make any difference which mode of treatment they received. But in contrast, for the individuals who had the high polygenic score for response to the environment, it made a huge difference. They did really well if they received individual treatment. They did less well if they received group treatment, and they actually did really much worse than we would expect if they had parent-led treatment. Now, this finding requires replication, and we would then need to test whether it was useful, and at the moment there's a lot of work being led by Rob's group to do that, but it gives you an indication of the potential of this kind of approach. So what we really need to do now is increase power. I explained to you that after several years of working with many leading clinical academics, in the end, in my data set, I had less than 1,000 subjects, so really not ideal. So we've gone about thinking about increasing power in 3 ways. First of all, doing a meta-analysis, not just of our sample, but with other available data sets. Then thinking about using the primary care possibilities that we have in this country with IAPT, and then an experimental approach. And I'll tell you very, very briefly about each of those. So first of all, in terms of increasing the power, and this work was led by Chris Rayner, one of my PhD students, we had our sample of 1,000 children, and from 3 other groups at Ruhr University Bochum, University of Würzburg, and Karolinska, we obtained a sample of around 1,000 adults as well. It wasn't a particularly well-powered analysis because it's still really too small, but we did get a tentative heritability estimate of around 15% from this analysis. This is the Manhattan plot for the genome-wide association analysis, and then this is the gene-level analysis that I showed you earlier. So a hint of something here, but the sample really is too small. But if we go back to that figure I showed you earlier and we slot our 15% heritability for CBT response in there, you can see it's very much in keeping with what we would expect. We're just not as confident in that figure as we'd like to be, so we need larger n's to get really clear about that. So that's why we're doing these 2 further pieces of work. So first of all, our primary care approach, we are recruiting through the IAPT, Improving Access to Psychological Therapies initiative. We have an online recruitment resource, which we're rolling out in SLAM, South London Maudsley, which is the trust where I work. At the moment, individuals are signing up and providing consent and some brief clinical information. They're then completing a saliva kit through the post from which we can extract DNA, and all of that is then going to be used to help understand the change in their symptoms during the course of their treatment. Those of you not familiar with IAPT may not be aware, but you actually get weekly ratings of 3 core measures of anxiety, depression, and basically impairment, so capacity for work and how well your social adjustment is going. And those are all assessed weekly, so we can use what they tell us at the beginning about their demographic situation, their clinical symptoms, and combine that with their DNA and understand change over time in these symptoms. And these are just some screenshots from the website. The study is about to go live. This is a really nice animation we've had built to tell people about taking part in the study, which is linked with our bioresource at SLAM. So it also tells them about taking part in a bioresource study and being recruited into such a resource and then accessing further studies. So there's various pages that people can look at and frequently asked questions. And IAPT sees 600,000 patients a year, and we have funding for a year at the moment from NIHR to expand this study beyond SLAM nationally. So hopefully in a year's time we will have a starting point of at least several thousand having taken part in this study. And then the second study we're doing is taking an experimental approach to this question. My student Kirsten Purvis and I have been working on creating a mobile phone app which delivers a fear, learning and extinction task. And extinction is the process whereby individuals lose their fear of something by gradually being exposed to it in a safe, non-aversive way. And this is a very standard paradigm that's been used in psychology for over 40 years, usually to detect case control differences between anxious case groups and control groups. We've developed the methodology to use to assess individual differences across the full range, because that's what we do in genetics. We're interested in individual differences. And we're going to be using this app to collect data in our twins early development study, which is a sample of twins that we've been following for about 25 years now. They're in their early 20s, and we have longitudinal data on them throughout their lives. And also in ALSBAC, the Children of the 90s study based in Bristol. And we collect ratings from individuals about their fear learning, and we will then be able to do twin analyses and also genomic analyses to understand the extent to which genetic influences are important in the development of both fear learning and also fear extinction, both of which are relevant to the development of anxiety and the treatment of anxiety. So where could this type of work lead? Well, my dream is that by the time some of the younger members of the audience are reaching the ends of their careers, perhaps even sooner than that, somebody comes into the clinic, into their GP perhaps even, and they are presenting with anxiety or depression, and their GP or their clinician knows that they have this wide array of possible treatments. They could enroll them on an online course and they could do that almost immediately. But it doesn't suit everyone and it's quite hard work, and it's a bit silly to allocate somebody to that if it's not going to be much use to them. They could put them on a waiting list for one-to-one psychological treatment, but that could be quite a long wait, and maybe they won't be able to manage that, or maybe there's something else that could be done sooner that would be more helpful. They could prescribe antidepressants, but it's difficult to know which one to prescribe and whether that's the right thing for this person. And right now, we really don't have enough information to be able to tell our clinicians how to make these choices and how to advise their patients in making these decisions. So my hope is that in the next 10 to 15 years, by combining clinical questionnaire data, demographic data, and genomic data, and perhaps other types of data as well, we will come to a point where we will be able to share likelihood of positive outcome for individuals following different types of treatment. So this kind of work doesn't take place without help from a lot of other individuals. These are all the teams that took part in the child anxiety Therapygenetics study, and this was the team that took part in the meta-analysis. This is the team that are launching the IAPT online portal with myself, and this work is being done particularly with Jerome Breen, also at King's with me. And I just wanted to end by telling you about our blog, because I feel really strongly about getting the sort of work we do out to a wider audience. And if you want to be able to read something really quick and easy and you don't want to have to read papers all the time, we put out a couple of blogs each week. We just last week started an A to Z blog, so there was A for anxiety and B for biobank. This week it's C for CBT. You get the idea. And we have 3 other main categories. Research Matters, where we summarise research findings from our own group and from other people's work that's of interest to us. We have a category called The Wider World, where we reflect on activity going on outside the research community. For example, I wrote a piece on Children's Mental Health Week, just bringing to people's attention how young many of these disorders begin, how early. And then this final section, which may be of interest to some of you, we call Life Scientific, and this is about our lives as researchers. And I've written lots of pieces about life as an academic, as a mum of young children whilst trying to be an academic, and I think some of those balancing jobs can be quite difficult for people, so there's a lot of pieces about that and about different stages in a career, research career, on this section of the blog. So that's my Twitter handle and my email, and I'm really happy to be press ganged by anybody to answer questions at any stage. Thank you.